So hello, everybody. Welcome to this month's Heart Muscle Talks. My name is Perry Elliott. I'm Professor of Cardiovascular Medicine at University College. And today we have a great presentation from Yolanda Van Der Velden from Amsterdam. And she'll be introduced by Lucy Carrier from Hamburg. Before we get into the main part of the meeting, I'm just going to hand over to Alex Protnotarius, who will just give us some instructions on how to use the platform today. So Alex.
Yes, so essentially this is a in a webinar function. So all the attendees can either post a question or comment in the chat, or you can do it under the Q&A button, and there you can also post anonymously if you want. And if you want to speak up, I can give you the right to do so. And please raise your hand if you want to do that.
Great.
So
thanks, Alex. So Lucy, over to you to introduce our speaker for today.
Yeah, so hello everybody from my side as well. It's my pleasure to introduce Yolanda Van Der Velden. I think we try to find for how long we know each other, but I think it's almost 20 years. For 20 years, and in a way, she's my little sister on genetics of hypertrophic cardiomyopathy. So Yolanda is professor of physiology, and she's the head of Department of Physiology of Amsterdam University Medical Center. And today, she will present a seminar on the state-of-the-art human models to test effectiveness of old and new drugs for inherited cardiomyopathies. Yolanda, the floor is yours, and I'm looking forward for your talk. And I will take all the Q&A questions at the end of your presentation to share the discussion.
Yes, thanks a lot for the kind introduction, Lucy. So, yeah, it's fun to be here. The title sort of, I had to think again, because I saw that many people link human models with the stem cell models, but I actually want to pull it a bit broader because I think a human model is basically any experimental model that mimics characteristics of, in this case, human cardiomyopathy, of course, the topic we are all very much interested in. And so what I want to do is sort of highlight this recent manuscript of two working groups of the European Society of Cardiology, where we sort of try to make an overview of all the animal models and animal-free innovations which are out there currently and which can be used to study the very diverse forms of cardiovascular diseases, among which is the cardiomyopathies. And so what we basically did is make an overview of what we now think are the models which are around to sort of study the different aspects of cardiac development, but also cardiac disease. So obviously human and laboratory animal models can span the whole range, moving from the cell level to even behavior, and of course organ-organ interaction and in vivo studies, aging. And if we move down, a lot of efforts, of course, are put into developing the stem cell derived models, and there are other cell models. And currently we think that those can currently mostly be used to study cellular pathomechanisms and also test the toxicity and effectiveness of drugs. And I'll touch upon this at a later stage, where I think really important advances have been made during past years. And but as you can see in this scheme, still a lot of the complexity which we would like to capture is not captured by the current human stem cell models yet, but the field moves on really quickly. So yeah, so what are the models to really model human? Which is, of course, it's difficult to find the optimal model, but it also relates very much to the question we try to address. And so I was to point out some of the models, the mouse models, which have been, of course, very relevant to study cardiomyopathies, and I'll focus here mostly on hypertrophic cardiomyopathy. And what we summarized, is currently the overview of animal models which are used to study the inherited cardiac disease. I highlight the different levels which were made during past years to use mice. So of course, we went from targeted deletion or overexpression of a gene, so you could identify the relevance of such gene, then to transgenic animals overexpressing a mutant protein. So this was a further refinement of models. But nowadays, having CRISPR-Cas, we are able to introduce a gene mutation, and we are actually able to mimic heterozygous and homozygous mutations as really as we are presented in cardiomyopathy patients. So I think this has been really an important advancement in having the different mouse models as a model to mimic human cardiomyopathy. So one example I would like to highlight is, for instance, the mouse model, which is used to model the founder mutation of phospholamban, which is very frequent in the north part of the Netherlands. This is a model made by the team of Rudolf de Boer. And basically what I like about is that they age the heterozygous animals, so up to 80 to 20 months. And then, like in the human setting, the disease phenotype is seen. So many of the previous mouse models, which are used to model cardiomyopathies, only show a full-blown disease in homozygous mice. But of course, the true phenotype, which is seen in most of the patients, is the heterozygous. And basically, it means that we need to spend a lot of time and money to wait for the phenotype to develop. But then this model showed beautifully that arrhythmias can be induced. There's impairment of contractility, remodeling, including fibrosis, and they also see the phospholamban protein aggregates. So there's really an important advance in having models which can be used to test drugs and to see which stressors may modify disease development. Now, another important topic which I would like to highlight is the biobanking of human cardiac sample, because I think that has been key into identifying important pathomechanisms and also drug targets, which eventually may end up to be used in the clinic. And my great example there is Chris Dos Remedios. He started the Sydney Heart Bank already in 1989. And this basically shows the whole map of the world where the many people who have used his tissue bank, which has been really of tremendous importance. And one of the remarks is that there's a quality control, and Chris really took care to collect all the samples in a very well and consistent manner. So the quality of these samples is very high. And I think he was a visionary in this respect. And we were lucky to collaborate with him. So using his well-preserved non-failing donor samples, we were able, for instance, to do a proteomic screen together with myectomy samples from hypertrophic cardiomyopathy patients, which we collect in collaboration with Michelle Michels from the Erasmus Medical Center. And basically to try to find new candidate proteins which may end up as a treatment target in the future. And just to highlight, which got a lot of our attention, also in collaboration with Lucy, is that we found that there's this upregulation of the microtubule cytoskeleton proteins. Zooming in, we found very high levels of D-tyrosinate tubulin. And this caught our eye because sort of for a couple of years, the team of Ben Proster reported on microtubule changes in end-stage failing heart. So he performed a lot of studies in well-preserved explanted heart samples and showed that there is an increase in detyrosination as well. And this links both with systolic and diastolic dysfunction of the heart muscle cells. So finding such an enormously elevated level of detyrosinated tubulin in hypertrophic cardiomyopathy, it's triggered us to look further and to figure out if this may be of also functional relevance. So also here we step to a mouse model, finding first the change in the tubulin setting, and we have the most frequent founder mutation in myosin binding protein C in the Netherlands mimicked in a mouse model. And we are now still currently doing the aging studies, but we use the homozygous mice to see if we could find changes in the tubulin pattern. And so using this model, we actually did find the same as we observed in the human myectomy samples, that there's a really enormous increase in detyrosinated tubulin, which would be linked with impaired contraction and relaxation of heart muscle cells. Now, having this mouse model then allowed us to do functional studies at the single cardiac muscle cell level. So then using a system where we can basically do high throughput measurements in cells which you isolate from the mouse heart and study basically all cardiac myocytes which we isolate with the system which is developed by Cytoskeleton, we were able to show that in this model, so these are the mouse cell, the myosin binding protein C founder mutation compared to the wild type, that there's basically impairment in contraction time, but also a delay in relaxation, so impaired relaxation. And if you treat the cells with so-called PTL, so partenolide, this is sort of able to reverse this detyrosination, so to bring it back to the low level. We could see that this intervention brought back the contraction time and the relaxation time to the levels found in wild type animals. So going from the human biobank, the tissue samples, and then using the mouse model, we got the evidence that detyrosinated tubulin may be a target to modify cardiomyocyte dysfunction. Now, another thing or another mechanism which really popped up during past years is at the level of the interaction between the actin filament and the myosin heads. And this also originated from studies in human samples. But I'll first introduce this concept because basically the myosin heads can bind in different states to the fin filament. So this would be the calcium mediated interaction. So when you have an increase in calcium in your cardiac muscle cells, it binds to troponin, and binding sites for myosin are released on the actin filament. So the myosin heads will bind, and this state will consume quite a lot of ATP. So for each power stroke, ATP is hydrolyzed, and also for the detachment. But then what was identified only 10 years ago is that there's actually also a very low energy consuming state, sort of, which we think is sort of, protective for the heart. And this is the super relaxed state of myosin. And this is a state where myosin heads are sort of interacting together and connected to the tail of myosin. And if you look at the energy consumed by this, if you would have 100% of the super relaxed state, you can see that ATP consumption occurs at a very low rate. So it hardly consumes any ATP. And then you also have the disordered state of myosin, which is in between, and that's consuming sort of in between the calcium activated state and the super relaxed state. This would be the black line would be sort of the combination present in heart muscle tissue. And the idea is if you would move between the super relaxed and the disordered state, that this would alter the energy consumed by the heart muscle cells. So if you would move from super relaxed to a disordered state, you would end up consuming more energy, more ATP. Now, this mechanism was further studied when it was found that it was first identified in skeletal muscle, then it was identified in heart muscle. And then the team of Christos Remedios actually also start to look at hypertrophic cardiomyopathy samples with, in this case, mutations in myosin binding protein C. And they actually showed that the proportion of super relaxed myosin heads is significantly reduced in the presence of myosin binding protein C mutations. So basically, this was proof using well-preserved human cardiac tissue samples that this shift from super relaxed state to a disordered state, and thus a higher ATP consumption, is present in myectomy samples from patients with hypertrophic cardiomyopathy. Now, then a good thing to work with would be, of course, also the stem cell derived models. And because we have the CRISPR-Cas method technique, you could also use these cells and introduce mutations. So I looked up, and we had a meeting a couple of years ago in Amsterdam, and this time, at that time, Thomas Eschenhagen, Christine Mummery, and Björn Nolman wrote a really nice overview of the status of the iPS cardiomyocytes at that time. And so a major thing is, of course, if you look at the normal situation, when you go in cardiac development, you have a period between 3 months of age and 70 years. There's a cardiac development and remodeling, and we know we have these beautiful rod-shaped adult ventricular cardiomyocytes. But then you have the stem cells, which then are differentiated in a dish for up to a month, and they look really pretty different. So what's needed to make them more adult-like? And I do think that the field advances really quickly, and we can already address a lot of questions. And it really depends on the functional readout, what you want to measure. But going back to this super relaxed state and the disorder state, so this model, the iPS-derived cardiomyocytes were used by the team of Christine Seidman, and they started with healthy pluripotent stem cells and introduced different mutations. And in this study, they introduced different mutations, heterozygous mutations in myosin heavy chain, and also studied this super relaxed and disordered state. And this shows the wild type. And what can be seen is that the super relaxed state also in these iPS-derived cardiomyocyte is reduced in the presence of a myosin heavy chain mutation, and while the disordered state of the myosin heads is increased. Now, and they used the myosin inhibitor known as mavacamten to see if this shift in myosin heavy chain state is corrected by the myosin inhibitor. And indeed, they find that mavacamten increases the super relaxed state and decreases the disordered state. So this also again beautifully shows that you can observe this change using human cardiac muscle tissue, but then also you can complement such studies with iPS-derived cardiomyocytes and see if the mechanism is present and if you're able to correct it. So this basically shows what's happening. You have this balance between the super relaxed and the disordered state, and mutations will shift it towards more disordered myosin heads, so a higher ATP consumption, there will be an increased contractility, impaired relaxation. So it can explain all the characteristics seen in the hypertrophic cardiomyopathy patients. And then what several studies now have shown is that if you then add mavacamten to the system, you move from the disordered myosin head state to the super relaxed state. So these have been really wonderful studies building up on the mechanism and showing that mavacamten is able to to lower ATP consumption. And of course, Jacopo reported on the positive trial with mavacamten. So just to summarize, just these two observations, if you use human models, I mean, our team has identified that high detyrosination tubulin levels are present in myectomy samples of hypertrophic cardiomyopathy patients, and we combine it with the mouse model, and the super relaxed state change and the effectiveness of mavacamten has been shown using human cardiac tissue and using iPS cardiomyocytes. So these were iPS-derived cardiomyocytes where mutations were introduced on a healthy background. But of course, you can also start off with cells which are taken from the patients. And I just want to highlight one of the studies. There are several now reported where they really can mimic differences in the cardiac phenotype in a dish using patient-derived cells. And this is a study from my colleagues here in Amsterdam, Rico Houtkoop and Connie Betsina, where they looked at the electrophysiology of iPS-derived cardiomyocytes, and samples were taken from patients with a VLCAT deficiency. So this is a recessive inborn error of the metabolism. There are really diverse mutations in a gene which is involved in fatty acid oxidation. So basically, the problem really starts at the level of the mitochondria and leads to lower long-chain fatty acid oxidation. So what happens basically is that this whole chain of breakdown of long-chain fatty acid is disturbed. You get a buildup of acetylcarnitine, and less energy is available due to these mutations. So what they did is they started off with the patient cells then and did the reprogramming. So you have then pluripotent stem cells, which you can then differentiate into cardiomyocytes, and you can hopefully model the disease in this way. And they used tissue or samples from two extreme patients. So patient one has basically a mild phenotype, a mild cardiac phenotype, while patient two had a much more severe cardiac phenotype, dilated cardiomyopathy, and really a difference in VLCAT activity. So they really took extreme phenotypes and then used and made the cardiomyocytes from these. And if they then looked at the action potential, they showed that it's shortened in both cell lines compared to a control cell line. So it's abnormal electrophysiologic signal for both cell lines. And they also looked at delayed after depolarizations, and they were also much more, or they were very frequent in both patient cell lines compared to the control cell line. But then they wanted to see if they could rescue the electrophysiologic abnormalities by adding resveratrol, which is linked to sirtuins, which are supposed to improve the mitochondrial function. And using this intervention, so potential drug intervention, they then found differences in the response between these different patient cell lines. So the shorter action potential in patient 1, so the less severe patient, was basically rescued by adding the medication, while this was not the case in the patient with a more severe cardiac phenotype. So I think this shows the potential of even using the patient's own cells and to show if an intervention will be effective or not. And they also did again or looked at the delayed after depolarizations and also showed that this was significantly lowered in the patient cells from the milder patient, while cells from the patient with a severe cardiac phenotype did not respond. So then, sort of, where are we? In this recent paper, we thought of really what's needed to further improve the stem cell derived cell system. So we'll be able to really use them for more purposes. And these are several things which I'll highlight. So reduce experimental variability, improve protocols to improve maturity, and finally also improve functional readouts. And this is a lot of text, but I'll go through it because I do think there's a lot to gain to reduce the experimental variability. And that's really where we can do a lot with the community to really exchange our protocols and really try to make standardized procedures. What should be done is really think about defined passage number, prove that there is a normal karyotype, identify high pluripotency markers, and that there is really a repetition in the experiment. So for each cell line or patient line, have 3 batches from 3 different lines. And there are already really wonderful initiatives. So there's actually a biobank of pluripotent stem cells in a registry. What we ourselves in the Netherlands started is a journal club to exchange protocols and to have a community of the younger scientists, so that they are not all alone trying to set it up themselves, but share their experiences. And I'll come back to this automation and high throughput facilities to really measure functional properties or metabolic characteristics of the cardiomyocytes will be key, in particular for drug screening, and I'll come back to that. Of course, I think everyone recognizes that the major limitations is that it's really costly to start it up and then to maintain not only material-wise, but also all the people who have the proper hands to do this work. Really needs major investments. That's one of the beautiful studies I would like to highlight is from the team from Florence together with the team in Seattle, where they applied a nanopattern surface to improve maturity. So if you really want to look more carefully about what happens to the action potential of cardiomyocytes, what they showed, if you plate the iPS-derived cardiomyocytes on a nanopattern surface, you end up having these beautiful rod-shaped cells, which really truly look like the brick-shaped cells, which we isolate from the heart. And this further shows that, for instance, they looked at the T-tubules and they really show a beautiful patterns. And that is really, this intervention really increases maturity of the stem cell derived cardiomyocytes. This is one example to improve maturity of the cardiomyocytes. This is a recent paper from the team of Björn Nolman, also looking at, in this case, the characteristics, the electrophysiologic characteristics focusing on the potassium channel. I won't go to it, I just want to refer to this paper. And here they incubated with dexamethasone and 3-iodotyrosine, and showed that this really helped to improve the maturity of the cardiomyocytes, changing the action potential to a more mature-like feature. So many ways, and then of course also the one which is well known is growing the cardiomyocytes in more complex structures, the 3D engineered heart tissue setup introduced by Thomas Eschenhagen, which also very much helps to improve maturity of the cardiomyocytes. So sort of, I think what's important is that we are working on reducing the experimental variability. We have different ways to improve maturity. So focusing on this part of the differentiation or the whole protocol. I do think it's really important. I hope there are a lot of medical doctors that biobanking is key. So what we need to compare our IPS models to is well collected cardiac tissue samples from patients, but also, of course, to start collecting patient-derived stem cells, which we will be able to use for all future studies. And then to come back to the title, State-of-the-Art Human Models to Test Effectiveness of Drugs for Inherited Cardiomyopathies, we have the therapeutic screening. And I think there it's also important advances have been made. And it's all to do with improving the functional readout and having an automated high throughput system. And I think we really made important steps there. I'll show one which we worked on together with Cytoskeleton. So I show that we can do functional studies in isolated single cardiomyocytes from animal hearts, but we are now also able to do such measurements, high throughput in 2D cardiomyocytes. So you can just see that you make continuous movies from the calcium transient and shortening of the cardiomyocytes and get a lot of useful information. Basically, we can monitor the main characteristics of the cardiac muscle cells. I myself, starting off with studying the really beautiful brick-shaped cells isolated from human heart preparation, could hardly believe that we could get useful information from these 2D iPS-derived cardiomyocytes, but I actually am convinced that we do get important information out of this system, both on calcium transients and contractility, because we actually do see what we would expect on the basis of different mutations which we introduce into the system. So what we've been doing in the past couple of years is that we use, in this case, troponin T mutations, which associate with either DCM or hypertrophic cardiomyopathy. And we've performed many of these experiments with 2D or iPS-derived cardiomyocytes in a 2D setting. For instance, with the HCM mutations, we find a delay relaxation, so we find an impaired relaxation. And we checked the response to beta-adrenergic stimulation with isoprenaline and see, for instance, that the DCM mutation does not respond anymore. So we're still validating this system, but it is very powerful to get the proper or many relevant and functional parameters out of these stem cell derived cardiomyocytes. And another thing which is recently published, or actually next month, will be is Björn Nolman's team again. They used HEK cells, human embryonic kidney cells, where they introduce a potassium channel variant, which is associated with long QT syndrome. And they developed a way to sort of see how well this channel still responds using a thallium flux. And this also provides a platform to do compound screening. So I think really many steps have been made in developing these high throughput platforms to test or to do compound screening. So really, if you think about the question, are these patient cells, can you test the effectiveness of drugs for inherited cardiomyopathies? The thing I think you should really think about is what's the functional readout that you need for your compound screening. So I believe currently that even fetal-like models may be of use as long as you know which cardiac phenotype you want to model in your IPS system. So if you are really looking at improvement of relaxation parameters of your cardiac muscle cell, then it may be already a proper model to do a first step in compound screening using these models. But one thing I always end my talks when I talk about the advances made in the stem cell research is that we shouldn't promise too much too fast. And this is Dutch, but it says, it's a newspaper article from 2018, and it says a new drug for ALS, a step closer by. And I think we should be really careful. I know journalists always want to make very popular titles, which we as a scientist tend not to like because we are very careful with promising too much. And it was about a beautiful paper where they developed a 3D model for ALS, so using stem cell derived skeletal muscle. But if you looked into the paper, this model only developed or even less than 1% of the force developed by muscle which you would isolate from a human skeletal biopsy. So I think we are making great progress, but like with many studies which have been judged in mice, I think we should be careful and not make the same mistake so that all we do ends up, well, too quickly in what's this? At the bottom of the, I forgot the English name, and doesn't end up in the clinic. So that's what I would like to end my talk with. Again, coming back to this paper, I hope we keep stimulating an open discussion about the models for cardiovascular disease, so we can all only make each other, doing even better. Thanks.
Thank you very much, Yolanda, for this beautiful presentation. This was a comprehensive analysis of what we can do from human cardiac tissue to human iPSC, derived cardiomyocyte models, including the use of different animal models. I think it was very good, excellent even. And also with all the care you took at the end to say that we have to be careful and also continue to work together. I think I completely agree with this. Okay, so for everybody, you can put your question in your Q&A. You can raise your hand. I will share the discussion, and I open now the Q&A. Oh, where does it go here? Q&A, okay. Okay, so we have already two questions, and I will start with the one of Jacopo, except if Jacopo, you want to tell it live.
Sure, thank you. Yolanda, congratulations. Can you hear me?
Yeah, I can hear you.
Okay.
Thanks.
I enjoyed your talk. Well, of course, I'm very curious about your view on myosin inhibitors, because we have used them, these drugs as clinicians, and I must say the short-term results are very encouraging. But what is your thought about the very long-term use of these drugs in a young patient, for example, who would need to be in the drug for a long time? Do you foresee a continued action or for example, escape mechanism which would limit the use? And what do you think is the disease modifying potential based on what you know in downstream calcium regulation and so or whatnot?
Yeah, so and I'm very much obsessed in past years by mitochondrial dysfunction. So related to studies we've done, also using imaging protocols, we see that even in mutation carriers who do not have remodeling of the heart, there's relatively high oxygen consumption of the heart muscle. So in continuation of that, we have done now a lot of studies looking at mitochondrial function, and that seems to be very much impaired, both we have a separation in the samples with and without a genotype. So and what's interesting is that in sarcomere mutation positive myectomy samples, the mitochondrial dysfunction seems to be very much linked with the mutation and not so much with the remodeling, while with the samples where we know there's no sarcomere mutation, so the sarcomere mutation negative group, it correlates very well with the amount of remodeling, so the septum hypertrophy. So there it appears to be more linked to remodeling. And my guess or a hypothesis would be that if you use MAFA, you sort of reduce the burden which is put on the mitochondria. So in that respect, my hypothesis would be that MAFA would also prevent or maybe even reverse mitochondrial dysfunction and thereby also prevent remodeling or even fibrosis. So that's something we hope to explore in the coming months.
Thank
years,
you. That's encouraging. Thank you very much.
I guess Perry want to jump in.
Yeah.
As always.
On the same discussion on Biobanking.
Well, no, just to pick up on the point you've just raised, Yolanda, because one of the aspects about this and other phenotypes is that they're dynamic and that they change over time.
Mm-hmm.
And you raised the point of mitochondrial dysfunction. Well, obviously, the mitochondrial reserve, as it were, may change with time, as with the accumulation of mitochondrial DNA mutations and so on. And also, there may be other influences of aging on the cellular phenotype. As you and Lucy know very well. I mean, there's always been this mystery of the timing of development of left ventricular hypertrophy and hypertrophic cardiomyopathy, and particularly in myosin-binding protein C, that it's often quite a late phenomenon.
Yeah.
And I know you and Lucy have got ideas as to why that might be, whether it be due to deteriorating proteostasis with time. But how do we, or should we even try to model these age-related changes in the kind of in vitro systems that you're describing?
Yeah, in vitro. So we are now ourselves using the mouse model with the founding mutation, so aging. And we actually have the first results now analyzed, and mitochondrial changes appear to happen at around 9 months in the heterozygous mice, which sit still in their cage basically, or they don't, well, they run a bit, but so, and we have to see how it links with the other characteristics which we measure by MRI. The in vitro system, I like the engineered heart tissues are wonderful. I mean, that's really the one which we can keep for a long time and model for a longer time and add stressors. So that's really a good system to, I hope, also get more of an idea what's the sequence of the changes introduced by the mutation. Maybe Lucy has additional thoughts.
I think this question was asked to me already at the first heart muscle talk.
So this is yes.
Yeah. I mean, engineered heart tissue, you can maintain them long, but also in this 2D model, if you go use the nano pattern, you can maintain the cardiomyocyte for more than, for 100 days, and maybe you could see a change in phenotype over time. Maybe not in the beginning at 2 weeks of culture, but maybe with this long-term culture on nano pattern with also elongation of the cardiomyocyte, maybe this is the way to go, right?
Yeah,
Mm.
and I'm hoping we hope to set up cardiac tissue slices. Andreas Dendorfer developed a nice system. So we'll try to apply that to the myectomy samples and see if there may be some reverse remodeling still. That's, yeah.
Thank you.
Okay, so I continue in the Q&A, and there is a question from Jan Smida, sorry for the pronunciation, who said that they developed in Israel a method for reprogramming human adipose cells into cardiac cells. Can this technology replace cardiac tissue biopsy? This is a question. What do you think?
Yeah, but I understand that you basically have you start with another cell type, right? So like you can also isolate PBMCs from the blood tissue, which is now regularly done. So I showed an example where they started with the skin tissue. And basically you can indeed reprogram different cell types towards a cardiomyocytes in the end. Can it replace cardiac tissue biopsy? If you looked at the one example I showed for the patients with the VLCAT deficiency, I do think at some point it will be a good replacement. It's just that now the protocols really take a very long time, like 3 to 4 months to even end up with the pluripotent stem cells, and then you need to differentiate them to cardiac muscle cell. So the protocols just take a very long time, but one day, yeah.
What I could add on this is that iPSC, direct cardiac myocyte, is now the state of the art to do this. But of course, any other parallel method are, of course, useful to reproduce what we observe in patient myectomy. And another point which is important is that also with iPSC now we can differentiate into cardiomyocytes and into any other cardiac cells, including fibroblasts, endothelial cells, vascular smooth muscle cells, which are part of the heart. And this will be also a method to develop kind of organoids in vitro to be closer to the human situation. Okay, Katja Gemlich, hello Katja. She has also a question for you. She said, great talk. So the pain of her own research is why the mice are not showing a phenotype as the humans? What are the differences? In your experience, Yolanda, what is the best way of provoking a phenotype in mice?
Yeah, it's a good question, and I do there's another unpublished study together with the mouse of Lucy where we used the Western diet, and that I think is a good trigger of a phenotype. Many people use aortic bending. I don't like that. It's for me a too much all or nothing stressor, so I think it's rather unphysiologic. Beta adrenergic stress, I do like. So I think if you would want to really challenge them. But in the end, what I really like the aging, what I showed. I think that's maybe the best way to get solid data. I mean, the humans, they need to age before they show the phenotype. So, but I realize it's very expensive.
Good. There is also a question from Sabine Klassen. Hello, Sabine. Great talk, Yolanda. Do you think that there will be a difference in the various sarcomere mutation carriers in response to mavacamten? Will myasthenic chain mutation carrier respond much better?
I think so. I would expect so. There are many mutations also in the tail of myosin, and I would just, based on studies which have been done, looking at the behavior of the different myosin. So we have the myosin mesa, which is in the head region, where there's really this hotspot where myosin mutations alter myocyte function really differently compared to mutations in the tail region. So based on those studies, I would suspect that they would respond differently to MAVA. And also, but still, I mean, that's the question I always ask when Jacopo presents the wonderful data on the clinical trial. Also, the people without a sarcomere mutation respond beautifully to MAVA. So I love the studies, but there's more to it than only the correction of the mutation-induced effect.
So do you think then that mavacamten only work on the SRX?
No, I think like there must be other changes acutely and maybe on the remodeling part, but that's
Yeah, because actually just to continue on this, so I think for everybody it's understandable that mavacamten works on myosin heavy chain mutations, right? Because it changes the state of myosin. But how do you explain, or what are the mechanisms by which myosin binding protein C mutations alter the SRX? Do you have any idea on this? Do we know?
That's the studies which were done by the team of Christos Remedios is on the haploinsufficiency, and that if you have reduced protein level, you also shift it from
SRX to
your lower super relaxed state. But then apparently MAVA would also recruit myosin heads from the disordered state.
Mm-hmm. That's good hypothesis. Yeah. Perry, you want to step in?
Yeah, no, I think this is a really interesting point, and I see Jacopo's got his hand up as well, because at the moment the use of the drug is predicated on the fact that anybody with hypertrophic cardiomyopathy may respond, and to get the drug into market and from a commercial perspective, that probably makes a great deal of sense. But as I and many other people have said before, we really want this drug in clinical practice, but we do need to proceed quite cautiously, because the effects of these mutations, as you both know very well, better than I do, that they have different effects on actin-myosin interactions. Yeah, that's what's behind your question about myosin binding protein C, Lucy. So there may be circumstances actually where some of these mutations have a neutral effect, or maybe even already induce hypocontractility at a cellular level, and introducing a myosin inhibitor may have some detrimental effects in that circumstance. So I think this may play out with time. And when you were giving your talk, I was thinking to myself, we might get to the situation possibly where we have to do some in vitro testing before giving MAVA, perhaps? I don't know. I'm not sure that would be very practical. Jacopo, what do you think? Do we give it to everybody and see what happens, like we usually do in clinical practice, or do we try and do it in a more targeted way?
I agree with what you say. On the other hand, we were sort of expecting to see a good result in mutation carriers, even the protein C mutation. After all, you're sort of removing a handbrake on the myosin as well. So, but it's really amazing is the response of the genotype negative. So I agree with your word of caution. On the other hand, I think this is a good opportunity to learn more about genotype negative HCM, because if it really is sort of polygenic, if it really is sort of to some extent age-related, it may even have to do with age-related processes, mitochondria and other stuff. We know from proteomics study that there may be very similar proteomics irrespective of the causing mutation in HCM. So to think that even acquired phenotypes or polygenic phenotypes where the environment may play a role may respond to this drug, again, I think it's fascinating. And again, one of the things I often say is we need to learn to modulate the sarcomere, pretty much like we're modulating the coagulation cascade or the adrenergic system. So we need to think about the lessons to learn from here, from this sort of almost unexpected finding.
I do think it's intriguing, right? Why do we have all these super relaxed myosin heads? It's so in that sense, if you would, I mean, that's been proposed by the people who found it, basically this state, that it's actually sort of if you
Hibernation.
can use them more, it's actually beneficial, maybe beneficial for any or many different disease entities, not only HCM.
For example, what do you think happens with age? Do you think that with age, would you expect the supralax to be less and less? Because of course we do see an increase in ejection fraction with age, which is also related to the loss of longitudinal function in the heart. But in fact, we often elderly people, even healthy people, have a sort of similar, sort of a semi-hypercontractile state, if you want, which we always assume is compensated for the loss of longitudinal function. But why shouldn't it be that you may have the same sort of situation where the beta myosin is overactivated compared to younger individuals, and to some extent that maybe.
I think that could come from, I don't know. I don't think that has been studied, but I mean, that could be done using Chris's tissue bank and looking trying to figure out if there's an age change.
New point application.
Yes.
Yeah, and do you think we can measure in routine this SRX and DRX state of myosin, also in iPSC-derived cardiomyocytes?
Yeah, I think it's really a lovely assay. Yeah.
Yeah.
Yeah, it's quite doable.
Okay,
may I ask you, just you both, I mean, Yolanda, one of your questions was, what are the functional readouts that we're interested in? And I think that's a problem across the board from the in vitro work into clinical as well. And it's going to be increasingly an important question as new therapies start to come online or start to be evaluated in phase 1-2 trials. So what are we trying to achieve with therapy? Is it all about changing relaxation? Is it about reducing fibrosis? Is it about reducing the twitchiness of the cardiomyocyte? I mean, does the in vitro phenotype translate into a tractable human phenotypic trait which is targetable by therapy? I don't have an answer to this question, but I think I think it's a conversation I think we're going to be having more and more in cardiomyopathy land, because in heart failure trials it's all about sort of stopping people dying. But in cardiomyopathy land, the therapies we give, we're not going to have those hard endpoints. It's going to be much more functional. Yeah.
I think what's nice to see is that we have more and more high throughput assays to screen, to look at different components of the heart muscle. So I think that's, well, maybe at least that we can really get to your questions and try to model what you would like to test.
Once we've made up our mind.
Yeah, let me know.
So there is a question which is also interesting for you related to tubulin detarosination from Matt Stroud. Lovely work, Yolanda. I had a question about the effects on tubulin detarosination in your knock-in HCM mouse model. And
if the phenotype can be rescued, right?
Yeah, but using detarosination drug, I mean detarosination targeted drugs alone.
Yeah, I think Lucy can answer that better than I do, because you are a step. That's basically part of the research. That's and yeah.
Yeah, so I can tell something indeed. So we actually used, there is this is a cycle, detarosination and tyrosination. And as Yolanda showed, this detarosination level is high in hypertrophy, in heart failure in general, in FPEF, HFrEF, all model of heart failure in the heart. And you can reduce this by parthenoloid, as she presented, but you can also use the overexpression of the other enzyme, which bring back the level of tyrosination to higher. This is a tubulin tyrosine ligase. So we did use this in our similar HCM mouse model. It's a knock-in homozygous. And we were not able to rescue all parameters, and particularly not hypertrophy, but we could improve markedly the compliance and the PV loop, the diastolic filling. So there is another way to rescue the phenotype in this HCM mice.
Yes.
And there is also development of drugs to target the tubulin carboxypeptidase as well. But this is a work in progress. There is another question from Cristina Gil Ortuño, and impressive talk. Yolanda, what do you think of spontaneous animal model like feline cardiomyopathy, feline HCM actually? How can we take advantage of these natural models?
Yeah, so we also describe those in the consensus paper. I do think it's a wonderful model. I just think it's quite a challenge to follow them properly. So I'm myself not a veterinarian, or I know there's a team we collaborate with in Utrecht, and they really put a lot of efforts into setting up a pipeline to use these animal models. But for sure, it's a unique source where you can gain a lot from. Yeah.
I mean, there is a Maine Coon cat with a methionine protein C mutation. Yeah. The problem is that we also wanted to work on this to test gene therapy, but it appeared that even the wild type can develop some HCM phenotype. So this is very difficult to establish. Okay. And I can tell you that the owner of these cats, they don't want to share, they want to have control animals, and they manage to make the segregation to get only wild type animals. And it's very difficult to get them and to breed them. But this is a bit a problem, because meaning that in cats in general, HCM is a typical phenotype. And therefore, this HCM Maine Coon cat, it's one model, but even the control could also develop a HCM. So we have to take this in consideration as well.
Yeah, as you say, I mean, we're at time now, Lucy, but you're right about the cat. I mean, from a clinical phenotype, it's fascinating because it looks exactly like the human phenotype. Yeah. I mean, if you take the scale off and you put the echoes side by side, they look exactly the same. They get outflow tract obstruction, they get sudden cardiac death, they die from heart failure. But it's 1 in 30 cats has hypertrophic cardiomyopathy. It's very common. And as you say, Lucy, the classic genetic model is the myosin binding protein C variant, but the majority of cats with HCM don't have that variant. And do it experimentally, it's a huge problem because there are very few experimental colonies left now, because the Maine Coon cat is the size of a tiger. It's a huge thing.
Beautiful.
And they're very expensive colonies, and of course it's a long-term animal model. And if you take sort of conventional cats as patients, then genetically they're a bit of a mixture. They have very complicated parentage, but as a model for drug trials, it's probably underutilized. And I think rather than thinking of them as experimental models, if we think of them as patients, which is actually how the veterinary physicians think of them, actually as a model for testing drugs, it's probably underutilized, I think.
Yeah.
Anyway, on that note about the cat, Yolanda, I'd like to thank you. Brilliant talk.
I think fun.
It was really interesting talk, and I think stimulated a fantastic discussion. And Lucy, thank you for chairing so ably, and
it was my pleasure
for stimulating that as well. So I'd like to thank everybody for joining us today. We had a great attendance, and so please keep your lookout for the next talk, and we look forward to seeing you next month. Goodbye.
Thank you. Bye-bye.
Bye.
Bye, everyone.